Okay, so let's talk protein. All right, so last time we met, we discussed carbs, then we talked about fats, and then we got to discuss just a little bit about proteins. And we learned that they're made up of amino acids, which are put together in chains, bonds called peptide bonds. Okay. Proteins are quite a bit more complex in structure than carbs and fats. There's actually 4 different categories: primary, secondary, tertiary, and quaternary forms of protein, progressively getting more complex. A primary structure protein is pretty much just a simple chain of amino acids. If you look hard enough, you can see these little boxes would be one amino acid. So they're bonded together. Okay, like taking beads and putting on a necklace. Okay. More complex than that, there's 2 types of secondary structures, a helix, alpha helix, which is where that chain kind of coils up like a slinky. And then a beta sheet is where there's actually 2 strands, and they connect to one another by hydrogen bonds. And it kind of looks like, when you take a piece of paper and you make a fan out of it. And tertiary structures are where you've got combinations of alphas and betas there. And then the most complex, which is where hemoglobin belongs, is a quaternary where you've got several tertiaries. So a lot of folding and twisting, and a lot of that, like I was telling you the other day, is due to hydrogen bonds. Okay. Now, why am I talking about this? Because protein shapes are super important. Okay. Protein does not function correctly unless it is in its perfect shape. Okay, and something I would have used as an analogy. I love analogies. Okay, and this is what I call the slinky analogy, and it works really well because we got one protein that looks like a slinky shown here on this page. Everybody knows what a slinky is, right? Okay, slinky's a little toy, looks like a coil. Play with when you're a kid, okay, and it walks down the stairs. But inevitably, doesn't take long, a lot of times for a slinky to either get twisted up and get knots in it, or get stretched too far out, okay? And what happens at that point? No longer works right, okay? It doesn't fall down the stairs, doesn't walk down the stairs correctly. Same thing can happen to proteins. They can lose their shape when they get denatured. And that's what happens when the surrounding environment, as far as temperature or pH, changes, causing the protein to unravel, okay? So we'll talk about that a little bit more in just a minute. When we talk about enzymes, I'll show you just how sensitive proteins can be as far as that goes. Now, the other day we talked about what happens, or excuse me, the phrase, if something's happening in the body or getting done in the body, good chance it's being performed by a protein. And this demonstrates why it's so important for our body to maintain that body temperature so close to 37 degrees Celsius, or 98.6 degrees Fahrenheit, and why we have to maintain pH of our different body fluids to such a small and specific number. Like blood, we said we like to see it at 7.4. If we get too hot, too cold, or pH in our body gets off, proteins start to unravel, and then we lose those various functions, and then what happens to us? We're in bad shape. Okay. That's what denaturation is. A protein denatures. I always tell students, write this phrase. Denature means protein loses its shape, which means it loses its function. Proteins lose shape, they lose their function. Okay, one of our major types of proteins, and I'm going to talk about these in a little bit greater detail, are enzymes. Proteins that are called biological catalysts, which means that they are chemicals that increase the rate of a chemical reaction, which is the main object, or objective, I should say, not object. They are not itself changed at the end of the reaction and don't change the nature of the reaction or the final result. Okay? And it all relates to what we call activation energy, which is the energy required for a chemical reaction to take place. Think of it like a barrier. Okay? And I've got another analogy. Okay? In the last, I don't know, 10, 15 years, like Tough Mudders and those obstacle course type races have become pretty popular. And if you're not familiar with those, you run, 5 miles or whatever it is, and you have to do these various obstacles, climb over something, climb a rope, climb under something. Okay? So let's all pretend that we're running a race, okay? And we've got one last obstacle, it's a wall. Okay? We've got two choices. We can climb a 10-foot wall or a 5-foot wall. Regardless of the size wall, the finish line is right on the other side. If we climb the 10-foot wall, it's going to take us a little bit longer than the 5-foot wall, correct? Okay. What enzymes do is they lower that 10-foot wall down to 5 foot. And they lower the energy needed for us to go from reactants, the starting materials, to go to products, the ending materials. So it makes it easier to get from before the reaction to after the reaction. Okay. So the key phrases here is they lower the activation energy required. Make sense? Okay. All right, a little bit more about enzymes. Structurally, they consist of two parts, a protein portion called the apoenzyme and the non-protein portion called a cofactor, which may either be a metal ion like iron, magnesium, zinc, or calcium, or organic molecule called a coenzyme, which are usually derived from vitamins. All things that we get in a nice, well-balanced diet. Enzymes typically end with the suffix ase, A-S-E at the end, like ATPase and lactase and maltase and dehydrogenase and all these different enzymes that we'll be discussing at various points in the semester. There are a few older enzymes that, for whatever reason, they haven't changed the name to enanase. Pepsin, trypsin, and renin, we'll talk about at various points in the semester, are still kind of the old school terms. All those various enzymes of our body can be categorized into groups based on what they do as far as chemical reaction goes. And so I've listed these off. It's not something I'm going to really focus too much on as far as knowing. Dehydrogenases remove a hydrogen. Oxidases add an oxygen. Kinases add a phosphate. ATPases split ATP. Anhydrases remove water. Proteases break down proteins. Lipases break down triglycerides. Of those, we'll really kind of just talk about a few at various points. We're not going to really focus on so much the category that the protein or the enzyme is. More so, we'll just be talking about, when it's involved in a specific reaction. All right, let's look at what we call the lock and key mechanism. Enzymes operate via what we call lock and key mechanism, where an enzyme has a specific shape, specifically what we call its active site. Okay, the active site is a little pocket within an enzyme that is specific to what we call substrate. Substrate is just the fancy term for the reactant that fits in there, like a key that fits in a specific lock. Okay, we all have keys to your dorm, keys to your car. Okay, I've got like 10 keys on my key ring because I've got 3 vehicles between my wife's and mine and one of our old ones, and I've got house and several office keys. I can't just take my car key and expect it to unlock the door that we use to get into this room, because the key has a specific shape and the lock has a specific shape. Okay? Enzymes and substrates are the same way. Okay? For example, sucrose, which we learned is table sugar, has a specific shape that is only going to fit into the enzyme sucrase. You cannot expect glucose or maltose or something else to fit perfectly in here. And on top of that, when that substrate fits in that active site and binds to it, forming what's called a substrate enzyme complex, or enzyme substrate complex, sorry, I said it backwards. The enzyme actually kind of closes in on it. So it creates what's called induced fit, okay, to help keep it in there nice and snug so the chemical reaction can take place. And when that chemical reaction takes place, like we show here with step 2, we form new products, and those products are released, enabling the enzyme to free itself up so that it can do the whole thing over again. Notice we're showing that's a cycle of events, which if I go back to this, enzymes are not changed at the end of the reaction, is what I'm showing you here. Okay. Make sense? Now, what's cool is a lot of antibiotics and drugs actually influence the activity of enzymes. Some drugs actually can act like a substrate, meaning that they actually fit in there and they create a chemical reaction that doesn't require the actual real substrate. Some of them actually block that spot. So it's like they put a piece of tape over the lock and the key can't go into it. And so that's ways that drugs can influence enzyme activity. Something we don't really get into in this class. I wish we had more time because I'm not talking illicit drugs, but drugs and medicines are really cool, amazing things. All right, let's talk controlling enzyme activity. 4 things influence activity of an enzyme. The first one, temperature and pH of the solution, relates to that denaturing thing that I was talking about. Okay, heating an enzyme up may bend it out of shape, preventing it from properly functioning, demonstrating once again denaturation. Okay. What we show here in our figures at the bottom, this left graph is showing you the activity of enzymes and how effective they are. So the higher up you are here on the y-axis, the better the enzyme's working. Okay. On our x-axis, we're measuring body temperature. Okay. Body temperature normally is at 37°C, which if I draw an arrow up, actually is not the perfect temperature for us as far as maximal enzyme activity. So like when we exercise and our body temperature goes up a little bit, enzyme activity goes up, okay? But there's a point in which, and look how steep our graph is on both sides, if we get too hot or too cold, enzyme activity really, really rapidly diminishes, okay? Same thing for what's over here, but this is illustrating that the different enzymes of the body are found in different places. Like, for example, pepsin, which is found in our stomach, is adapted for a low pH because our stomach releases that hydrochloric acid we talked about the other day. Whereas trypsin, it's released in our small intestine. It likes a pH that's actually basic, somewhere around 8 or 9. Okay. Too high or too low, change in pH can basically stop those from working. Concentration of cofactors and coenzymes also affect enzyme activity, as well as concentration of enzymes and substrates. And then number 4 really relates to what I was talking about with medicine, stimulatory and inhibitory effects by non-substrate chemical substances selectively binding to the enzyme and altering its activity. All right, let's talk our final category of the organic compounds, what we call nucleic acids, which are the most complex, huge organic molecules containing carbon, hydrogen, oxygen, nitrogen, but also a 5th phosphorus. These are described as being key molecules for human survival, not that the other ones weren't super important. But why? Because they provide the directions for building proteins. Okay, we'll talk more about that here in our next lecture. But this is where your DNA and RNA are categorized. We've mentioned a couple times. DNA, which is short for deoxyribonucleic acid, forms the genetic material inside each cell in segments called genes. Genes are found within our chromosomes and code for the traits we inherit from our parents. Things like our skin color, our hair color, our eye color, which we are easy to see on one another. We inherit from mom and dad, and that's because of the protein synthesis that they code for. In this case, skin color and hair color comes from the melanin that we produce. RNA, short for ribonucleic acid, is a single-stranded nucleic acid, relays the information from those genes, from the DNA, to guide the cell synthesis of proteins from the amino acids. Nucleic acids are composed of monomer units known as nucleotides, and I'll show you what a nucleotide looks like here on this next page, which is an illustration of DNA. We are showing you up at the top in this box, one little segment of it. Okay, if I were to draw a box inside of this box right here, that would be one nucleotide. Here would be another nucleotide. Here would be another. So there's 4 nucleotides in this little box. There are 2 common components to each nucleotide: a phosphate, a sugar known as deoxyribose. That's a 5-carbon sugar, hence the name deoxyribonucleic acid. Okay. And then what's variable is what's called the nitrogenous base. In DNA, there's 4 different nitrogenous bases: thymine, adenine, cytosine, and guanine. Okay. Now, DNA is described as a double helix, and that's because there's 2 strands. Okay. And think of it like a ladder that's twisted. Okay. The vertical bars of our ladder, I'll call them bars, I guess, are orderly repeating arrangement of the sugar phosphate. Purple, blue, purple, blue, purple, blue, purple, blue, sugar phosphates. The rungs of your ladder, which you step on to climb up, and this is what causes the twisting, are hydrogen bonds that link A's and T's together, C's and G's. If you look at the little dotted lines, those are hydrogen bonds. Not something that I really get into in this class, but if you take biology, you would learn all about DNA in much greater detail. All right, let's talk ATP, adenosine triphosphate, which is kind of like a hybrid nucleotide hybrid sugar. It's a very important molecule composed of adenosine and ribose sugar attached to a string of 3 phosphates. So it's like one of these in that we've got a 5-carbon sugar. This one's ribose. Okay, it's got a nitrogenous base. It's only going to be adenine. There's no thymine, cytosine, or guanine. And it's got not 1, but 3 phosphates. Okay. Each phosphate has 3 negative charges associated with it. Okay, and going back to our lecture on chemistry, we said that opposites attract, right? Okay. Well, these are all the same charge. Like charges are not attracted to one another. In fact, they don't like each other at all. They repel each other, and if you look hard enough in our figure, right there and right there, they're actually showing the covalent bond with a little bit of a bend in it. Okay. Now here's another analogy. Okay. I grew up with 3 siblings, 2 older and 1 younger. Okay. Me and my little sister had to take turns riding in the back seat of our car growing up, in between big brother and big sister. And I don't know if any of you have siblings. Put 3 kids in the back seat of a car, there's a lot of friction. Okay, don't touch me. This is my space. I'm not touching you. You know what I'm talking about. Okay, especially if you have sisters or brothers. Yeah. That's what's like with these 3 phosphates right next to each other. So much friction and so much energy exists that the 3rd phosphate can actually be removed and transferred to something else, which powers it. Okay. Powers things like muscle contractions, movement of chromosomes during cell division, moving structures within cells, transport across membranes, and synthesis reactions, which we talked about the other day. Now, if that 3rd phosphate is removed, now we've got 2, and it becomes adenosine diphosphate. Now, it's not like that 3rd phosphate is just left and it will no longer play any role. We can pick it back up, adding that 3rd phosphate, and then we'll form ATP. And we talked about how this is an example of a reversible reaction just the other day. Last thing I want to talk about for ATP in this lecture is how we produce it, a process called cellular respiration. which we'll cover in much, much greater detail towards the very end of the semester. Two phases of cellular respiration exists. The first is called an anaerobic phase, which requires no use of oxygen. So that's why I've italicized the word without. This takes place out in the cytosol, where we go through what's called glycolysis. We form a small amount of ATP. The majority of our ATP is produced aerobically inside of our mitochondria, where we can net out 36 or so ATP, but this requires, and that's why I've put with, the use of oxygen. So you'll see the words aerobic, anaerobic at various points. Always know anaerobic means without oxygen and aerobic means with. Sound good?